It's just a cell, here's the nucleus, here's the DNA inside the nucleus of our own human cell. This is a human cell. So we make skin cells constantly, all the time, right? The cells have to replicate, and we always have to make new skin cells. So, and when we make a new skin cell, what's gonna go in there? You have to be able to put something in there. You have to be able to add more DNA. So anytime a cell replicates, it has to be able to make more DNA. And our cells, you know, even like our skeleton, you think, oh, we're kind of born with the same skeleton we die with. Every 7 years you get a whole new skeleton, a whole brand new skeleton. And that's because your cells are constantly replicating. So like, for example, I'm going off on a tangent, but for patients that are on chemotherapy. So patients that are on chemotherapy, what chemotherapy does is it attacks cells that are replicating quickly. So it will damage cells that are going through replication. And the cells that are going through replication are cells in the mouth. That's why patients that are on chemotherapy get mouth ulcers. Epithelial cells on the hands and the feet replicate really quickly, so sometimes they'll get damage to their hands and feet, but also the hair, the scalp, so that's why a lot of patients will lose their hair. So chemotherapy affects rapidly growing cells. So anytime a cell has to replicate, it needs to fill the hole, it needs to fill the nucleus, right? So it has to be able to copy. So same with a bacterial cell. So a bacterium, like let's draw a staph just for fun because we keep drawing E. coli. So, not a nucleus, don't do that. You have a nucleoid region and you have a genome. So what Staphylococcus aureus does is it asexually reproduces, which we're not going to talk about reproduction quite yet. I'll talk about that in a day or so, but they just reproduce. They just, you know, it's like if I were just to butt off and you'd have two of me here that were exactly the same. That's what bacteria do. So this bacteria, now it needs something in its nucleoid region. So now you have to have another whole set of DNA. So that's step one. Step one is it has to be able to replicate. Now you need 2 exact copies. Step 2 is what's called transcription and translation, which are the actual function of the DNA. What does the DNA do? So step 1, we're just going to make another copy. Let's start there. So how do we make another copy? So we make another copy during a process called binary fission within bacteria. And so DNA replication is controlled by a process called the cell cycle. But for that part, I want you to kind of put it in quotations for a second, because bacteria don't really go through the cell cycle like we do. And I'll tell you why. Let's forget about transcription and translation for a minute. So when we talk about replication, it's in this process called binary fission. So binary fission is another word for asexual reproduction. So asexual reproduction is what the point of sexual reproduction. The whole point is DNA compatibility, right? Mixing DNA so that there's an offspring. Mother in the egg has half the amount of chromosomes. Father in the sperm has half the amount of chromosomes. 2 of those have to come together to make a normal amount of chromosomes to produce an offspring. It's not the case in bacteria. They have all of their chromosomes in one spot, and they have to duplicate, they have to duplicate their entire chromosome again for their next offspring. They're still living. They're gonna replicate and go through binary fission. Replicate, go through binary fission. Replicate, go through binary fission. And it's just gonna keep making copies, spitting out new bacterial babies, right? So in the process, the cell will get larger. The chromosome will replicate, which we'll talk about in a minute, and then a septum forms. So not all bacteria will spit off a new offspring and it will be separate. So sometimes it'll get bigger and it will keep this little area between them. It'll keep this little area between them. Keep this little area between them every time it divides. What is this an example of? Streptococcus. Beautiful. So these are definitely individual bacteria, individual bacterium that are separate. Bacterium means one, bacteria means plural. But you separate them, if they'll either separate fully and go and live their own life, or they'll stay hooked together in a diplococci, a streptococci, a staphylococci, however they end up. So binary fission is just the copying. Yeah?
If they're not actually separate, do we think of them as being as 4 different bacteria?
Yeah, great question, because they can separate. They are individuals, because they can, then one will actually totally split off and go and start replicating and copying together. And, they can share things between, but they can also split off and go and live on their own. So, yeah, funky.
Sort of like Siamese twins?
Yeah, like Siamese twins, they can separate or they don't have to. Yeah, yeah. Yes?
So basically, it's only certain types of bacteria can connect, though.
Yes, depending on what this genus and species is. Like E. coli is never together.
Okay.
But like, is it Bacillus? Bacillus cereus? Is in a Streptobacillus? Don't quote me, I forget. I'll have to look at it. There is Streptobacilli, one that we look at, but I forget what it is. Yeah?
Binary fission refers to only when they are continuously attached or also when they separate?
Great question. Yeah, so binary fission is just asexual, it's just the reproducing and making new ones.
Okay.
Yeah, awesome. Okay, so let's talk about the replication process and how that works in bacteria. So what is the point? So I'm gonna reference this little grandma and her little recipe over and over. I just got this off the internet. But, so what DNA is like the recipe book. So there's a book, and this recipe book that's full of A's, T's, G's, and C's, right? If you were to write a book and write a 10,000 volume novel, it would just be A's, T's, G's, and C's. So in this whole chromosome, it's just A, T, G, C, C, G, G, A, but in a double strand, right? So this is the recipe book. And so what we need to do when we replicate the genome is to replicate that exactly for the next offspring. So how do bacteria replicate as part of binary fission? Binary fission, remember, is the actual asexual reproduction process, but in that process, we have to be able to replicate the genome. Okay, so how do bacteria do this? And why do we care? We care because we're going to, what are we gonna do with this process? Are we gonna do electrophoresis this? I can't remember, but you will need to know it, and it's physiologically important. Just hang in there with me. Why? So if you're supercoiling your entire DNA around here, the E. coli chromosome, there's one chromosome because of plasmids. We're going to talk a ton about plasmids. You'll need to know the anatomy of a plasmid. A plasmid is basically the same as the chromosome itself. So we're going to talk a ton about that. Okay, so let's look at the anatomy of the chromosome of bacterial chromosomes. Yeah?
Quick question.
Mm-hmm.
So we have human chromosomes, which look like X's. Chromosomes of bacteria are circular. In the DNA, is there multiple circular DNA, or is it one just giant circle?
Great question. So in bacteria, you mean, is it?
Yeah.
In bacteria, it's one giant circle.
Okay.
Yeah,
One
yes, so
continuous strand.
one giant continuous strand.
Okay.
Yes, yes, fantastic question. It's one just giant continuous strand in bacteria. Yes, and humans are so different in that way that ours are linear. So they're, you know, you have to have multiple spots and multiple things where bacteria just have to have one. Yeah, great question. Okay, so let's look at the anatomy of a chromosome, which is gonna come up again and again, so hang in there. So in the bacterial chromosome, there's a small spot called an ori. And this is called an ori, which is the origin of replication. So origin of replication in humans, we call it a Tata box. I don't know if you've ever heard of that, but a Tata box. And I don't care that you know that in humans, but the reason I brought it up is because I think it's cool, is because what does this remind you of? Yeah, tater tot box. What do those letters remind you of? Yes, adenine, thymine, adenine, thymine. So together in the double strands, that's what they would look like. Why would this be a good spot for replication to start?
Easy to break.
Because they only have double bonds. Yes. So here on our origin of replication, there's a lot of double bonding, there's a lot of A's and T's. Just throwing it out there. So that's called the origin of replication. So what happens is we're going to learn all about the enzymes and how this works. But what happens is we're going to have some enzymes come in and break apart those double strands. So we have to cut those hydrogen bonds, break apart those double strands, and start copying, and start making an identical copy. Okay, so just a little note about replication. All living things replicate their DNA. They have to, otherwise you'd have an empty cell full of nothing in it, right? 50,000 copies in an hour in E. coli genome. So in the time that we've been sitting in this class, our bacteria, their replication actually, because we put our bacteria in the refrigerator, their replication is way slower. All their enzymes are like dragon because they're cold. They're not working at their correct temperature. So, but normally when we put them in the 37-degree incubator, they'd be cooking. They'd be 50,000 more E. coli, 50,000 more E. coli, 50,000 more E. coli every hour. So they can replicate incredibly quickly. They have a circular chromosome. They have one origin of replication. So one origin of replication. So why would that be different to human chromosomes? I'm just going to go with traditional X's, how they'd see. So if we were going to replicate our genome, how would you just have one origin of replication? You'd have to have origin, 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 origin. Humans have a ton of origins of replication. Bacteria have one. So there's one origin of replication where all of these enzymes are going to come in and make a whole nother copy. Okay, so let's figure out how it does that. So there's a lot of little players in it, and these little players, we're only going to learn a small amount of these of the players of replication, but they're important. So replication happens bidirectionally. I don't care that you know what that term is. I think it's just obvious. It just goes both ways. It doesn't go one way and choo-choo train all the way around. It goes that way and splits until the whole thing's done. And so what happens is there's one copy, one of the single strands of DNA. Here's the other side of the helix, the double strand. So what's going to happen is at the origin of replication, all of these enzymes are going to come in to the origin of replication and start working around. And then at the end, those two little necklaces are going to come apart. And one, if you have a double-stranded necklace, now what it's going to look like is you're going to have here and here were your two original copies, because we split those two necklaces apart. Now you're going to have a new copy and a new copy. So that's what's called semi-conservative replication. So semi-conservative replication means we conserved one strand, we conserved the other strand, we made an exact replica, and now we have two strands each. So now you can put this in a new bacterial cell, you can put this in a new bacterial cell. Now we have copies. That's called semi-conservative replication. And one question I want to ask about that, this might be tricky to you, so let me just see if I can ask it in a way that's not silly question. So here's one side, here's the other side of the DNA double helix. Now, if that's gonna go into one bacterial cell, and this one's gonna go into the other bacterial cell, now we have this strand, and we have this strand. So, bless you. Does anybody else, oh here, let me use this better black. Okay, so if this one is gonna be copied, this is ATGCCCAAAG. We're just drawing it in a line as a placeholder. Here's all our A's, T's, GC's, A's, T's, GC's. So here's the opposite, T's, A's, GG, that will match up to those nucleic acids. So this black strand, even though it's just built, it's a brand new strand, will match what original strand? I'm asking that in a weird way. So if I'm not, I'm sorry, what?
The pink one?
Yeah, the pink one. So it's just making, all it's doing is it's coming in here and reading this exactly the way it is. But this one ends up just matching this one because they have to match, right? The A, if this was a, let me just give you like 3 examples. If this was a A, T, C, and here was a, what would be the opposite? So that was a TAG. So here you'd have an ATC because this one went to this copy, went to this cell. This one had a TAG. So this one would have a TAG, right? And this one would have a ATC. So this one actually just matched that one, and this one actually just matched that one. So it is conservative. It is, they match, that you have you conserved one whole strand, but this new strand matched the original strand. There's never a difference. It will be the same for 100 billion more years. We'll copy the exact same, copy, 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 copy. Yeah? Okay, kind of with me? That was just kind of a side point. Okay, so let's go over some of our enzymes. So our enzymes that you will need to know, and I have a little table at the end of all the enzymes you need to know. But one of the enzymes we need to know are helicase. So helicase is going to come into this origin of replication, and helicase I kind of like to think is the first enzyme, even though it's not really. There's a whole process. We're going to keep it as simple as possible to the things that I think are important. So we have an enzyme coming here that's called helicase. And what helicase does is breaks hydrogen bonds. So now it's easy to remember because helicase breaks H bonds. Helicase is going to come in and cut down the middle of the 2 strands and start with the cutting. Helicase cuts all the way around. You're going to have 2 helicase enzymes that are going to come and cut as you move along. So this topoisomerase is another enzyme, and we're going to remember that topoisomerase. So what does ACE mean?
It's an enzyme.
It's an enzyme. Yeah, remember like the little Pac-Man that comes in and actually does stuff? Topoisomerase untwists. So T, it untwists. Topoisomerase untwists. And as like how I gave you the example of the phone cord is, it's just gonna get twistier and twistier. You know, if you try to pull 2 necklaces apart, they're just gonna get bunched up in the corners. So it actually, I don't care that you know this, but it does actually break the covalent bonds and loops it back around so that the necklace will untwist. But topoisomerase untwists, that's what it does. It's an enzyme that untwists the double helix. Here you have, I don't know if I want you to know that or not, but you could just know it because it's not that hard. So single-stranded binding proteins. So single-stranded binding proteins, or sometimes they're called SSB, they hold, or let's say bind, because it's in the word, keep it easy for you guys to remember. They bind so that it doesn't twist again, so that they don't retwist back on each other, to keep those naked strands, because those, they're like glue. They're like chemical glue. The A's, T's, G's, and C's, they want to bind together very badly because they're chemically attracted to each other. So those single-stranded binding proteins hold those naked strands apart. Yeah?
The slide says single stand. Is that the typo or is that the typo?
I got this off the internet, but they're not, it's not single stand. They're
Okay.
single-stranded binding proteins. I don't know, that's a really good point. Yeah, I just got this off the internet like 3 days ago, so, but they are single-stranded binding proteins is what they're called. Okay, great question. All right, so, let's bring up one more enzyme and then we'll do our note card. So our last enzyme that we're gonna talk about for the day is we're gonna talk about DNA polymerase 3. What do you think DNA polymerase does? What is this word? Yeah, it polymerizes. It makes polymers. It pulls things together. So DNA polymerase 3 is important because we're going to go over what DNA polymerase 1 does. It does something totally different. But DNA polymerase 3, this is where all the magic happens. This is the enzyme that's going to be adding as it reads along. So it's going to be matching. So it's going to come up and read this strand. The DNA polymerase 3 is going to read this A and put in a T. It's going to read this T and put in an A. It's going to read this C and put in a G. So this is the polymerizer. This is actually the main enzyme that adds as it goes along. So that's what a DNA polymerase 3 does, is adds the nucleotides to make the double strand. So next time on Tuesday, what's today, Thursday? Gosh, I am all over the place. So let's have, for our next quiz, we're just gonna do the enzymes that we talked about today, replication that we talked about today, binary fission that we talked about today. What else did we talk about? On Tuesday, we finished with metabolism respiration. So we did aerobic. We're gonna do aerobic again. We're gonna do anaerobic. We're gonna do facultative anaerobes. What else? Is there anything else we talked about? Let me just pull that up really quick. Oh, we talked about exotoxins and endotoxins. We talked about, yeah, I mean, just exotoxins and endotoxins. That was kind of easy. And then
more in depth of like the aerobic metabolism, or like be ready for that, I guess.
of more in-depth aerobic
Like
metabolism.
in the last quiz.
Yeah, more in-depth aerobic metabolism. Yeah, so know the electron transport chain, know the pieces, but nothing that we haven't covered. So just, yeah, same as we covered. And then what an anaerobe is, that they ferment, and some of those byproducts like lactic acid, acetic acid, 2,3-butanediol was our neutral end product, and what was it? 2,3-butanediol, lactic acid, acetic acid, and alcohol, booze. Don't forget the booze. Okay. So then we'll talk about replication. So it's kind of a lot of funky things. So try and really pull it all together. Meet with Dr. Welsh on Friday. We can talk to Dr. Rosebeer if he's willing to get the group together. I can do a group, whatever you guys want to. If you're busy, I'm happy to do it. Or I'm happy to have a one-on-one with you anytime. It's a really good idea to watch videos. Watch videos on, especially on replication, and make sure that you can see it. So this replication video I have, just 2 seconds. I put a replication video on here. but it's way more detailed than you need to know. It's like a clamp loader and the blah, blah, blah. You don't need to know all that. Just keep your enzymes simple. Keep it, here, if you look on these same slides, we're gonna go through all this next week. Here is a list of our enzymes. Where's the list of our enzymes? Here, helicase, what it does. Topoisomerase, what it does. We don't need to know POL1 yet because we haven't talked about it. We talked about DNA polymerase 3. So those single-stranded binding proteins here, those were the ones that we talked about. And I'm happy to go over it again and again and again and again and again, a million times if you need it. Okay, so let's do our note card and then we will go down to the lab. So what we can do in the lab today is, I'm gonna have you guys look, what we're gonna do is we're gonna get do our cleaning, get all of our PPE on, and then we're going to set out our slides that we made on a little piece of paper. I can't think of paper towel. This is gonna be a scary day. Okay, paper towel. Slide on the paper towel, and we're gonna go around and look at everybody's. I just want to try that this time, and everyone's gonna go around and look at everybody's slide and just kind of get a sense of like how much bacteria is on there. Look and see how successful some people were or not, and no big deal. It's just all practice. But we're gonna get our slides out and look at them, and then we can look at them under the microscope, and then you can still gram stain again. And I would highly recommend gram staining again if you have time towards the end of lab. Yeah.
First though, are these like human cells, like bacteria?
Yes. Yeah. So it's these little guys here.
Oh, cocci, okay.
Yeah. So these are diplococci. So see how they're together? The diplococci. There's little tubes. Yeah. Great question. Okay. This is Neisseria gonorrhoeae. Let's say it 3 times together. Neisseria gonorrhoeae. Neisseria gonorrhoeae. Neisseria gonorrhoeae. This is gonorrhea. So last time we had chlamydia, which the way I can always remember the difference between gonorrhea and chlamydia, because chlamydia sounds like it has the word clam in it, and they're obligate intracellular. So they stay inside of a cell. That's what helps me remember. Chlamydia is way more common than gonorrhea, but gonorrhea is common as well. Chlamydia is actually there's actually a huge problem with chlamydia, and that thinking it affects fertility, and that people that once had it, it's affecting their fertility now, and that people that didn't even know they had it, that now, I don't know how much you know about fertility problems in the United States and around the world actually, but they're thinking, there's a lot of different factors, tons of factors, but one factor they're thinking also is that some people who had chlamydia when they were younger and didn't know it affects their fertility now. So kind of scary, but interesting to learn about.
And my friend had chlamydia, didn't know it, or she didn't get antibiotics for it, something like that. And
Yeah.
then she got married, was trying to have a baby, and she had scar tissue.
Oh,
chlamydia. And we're
freaking
in
heart
chlamydia,
tissue.
so it was really hard for her to have a baby.
and she did?
She was able to, yeah.
Oh, thank goodness. Oh,
But
that is so scary. I know fertility problems seem like, yeah, fertility problems are scary, but when and if, some of you already have children, but when and if you're ready to have children, you know how devastating that is, because really it's a huge part of life, and to not be able to experience that as part of life is heartbreaking. Okay, so the gray you don't need to write down. This is a gram-negative diplococci. So yeah, these ones can live inside of some of the white blood cells, but these ones are not obligate intracellular like chlamydia. That's how we were gonna remember that chlamydia. And remember, chlamydia is weird. We're not really sure about the peptidoglycan situation in chlamydia. They live inside of cells. We have to do that weird Gibbs stain. These we can gram stain, but we can also PCR. Do you guys remember, like, in some people, now a lot of people know, even though they don't know what PCR is or what PCR stands for, that they do know, are familiar with the term PCR because of COVID. But you can run a PCR, so it's hard to gram stain if those gonorrhea bacteria are living inside of a white blood cell. You can't gram stain a white blood cell because you're not staining the peptidoglycan, right? So sometimes it's hard to tell with just a gram stain, so you might have to do a PCR as a lab test. And PCR is polymerase chain reaction. We'll learn more about what that is. So there are many different species of Neisseria, and there's meningitidis. We'll learn a little bit more about Neisseria meningitidis is one of the bacteria that cause meningitis, but those are two different species. So genitourinary, sexually transmitted, and remember it can do the same thing, pneumonia and conjunctivitis, for its vertical transmission from the mother to the child as the child will pass through the birth canal. Less frequently, it can cause conjunctivitis, and I know it sounds gross, but it can cause pharyngitis of the oral pharynx from oral sex, and it can cause proctitis from anal sex. And then symptoms, mostly asymptomatic, which is terrifying, but also discharge, pain, urination, bleeding, infertility, and it can cause prostate cancer, actually.
So you don't want us to write down all the pink parts?
Yeah, no, that's just for if you don't have to write any of the less frequent stuff down. Okay, awesome. So go ahead, and when you get down there and get all your PPE on, then